Biology 107 Lab 9, Transformation of Bacterial Cells. If you remember last week, Lab 8, we extracted plasmid DNA from E. coli and then we analyzed it by agarose gel electrophoresis. This week, we're going to use the same plasmid and we are going to use it to transform some E. coli cells. Let's talk about what transformation means. As we discussed in class, we talked about Frederick Griffith's historical experiment in 1928, where he was looking at two different strains of Streptococcus pneumonia. So there are two strains, the S strain and the R strain, S for smooth and R for rough. The S strain had a carbohydrate capsule on it that made the strain pathogenic. So if you take a look at this diagram, the S strain is pathogenic and the R strain is not. So what he found is if you inject the R strain into mice, they're fine. If you kill the S strain by heat or other methods and you inject it into mice, the mice are fine. But the interesting thing that he found was that if you kill the S strain and add whatever is left to living R cells and inject it into the mice died. And from those mice, he was able to extract living S cells. So he said something must transform these cells, and therefore the word transformation came about. And in 1928, he did not know yet that DNA was the transforming variable. Skip forward to 1944, the Avery McLeod-McCartney experiment. They were using the same strain of Streptococcus pneumonia, and they basically took the Griffiths experiment to the next level. So what they did is they extracted all the different macromolecules using a little bit of chemistry, and so the proteins, RNA, DNA, carbohydrates, and lipids, and then they mixed those with the R strain. So they wanted to see which would be the transforming factor, and in fact the answer was DNA. DNA gave us the living virulent S strain. So they concluded that DNA was the hereditary material of the cells. So our experiment is going to be kind of similar to this one. We're not going to use a virulent pathogenic organism. We're going to use E. coli. We're not going to extract all sorts of different molecules, proteins, RNA. We're just going to use the DNA and confirm that DNA is the hereditary material of a cell. Here's an overview of experiment 9. So last week in experiment 8, we isolated and purified plasmid DNA from E. coli. So that plasmid DNA had as a gene on it that makes the E. coli resistant to kanamycin. So today's experiment, we want to see if we can transform cells that are not resistant, so they're susceptible to kanamycin, to be kanamycin resistant. So the way the experiment works is we're going to get two test tubes. There they are, 1 and 2. So test tube 1 is going to be a control, no DNA. It's going to have something called tris buffer. So the plasmid DNA is in the tris buffer. So test tube 1 has everything exactly the same, the tris buffer and everything, just not the plasmid DNA. So it's a control. Test tube 2 is going to have the plasmid DNA in it. Both test tubes are going to have E. coli cells, and they are going to be susceptible to kanamycin. So what we're going to do is mix them together, go through an experimental protocol, and try to see if one of them will uptake the DNA and become transformed. So after that, we're going to put them on agar plates with kanamycin. Test tube 1 is a control. It should not grow on the kanamycin. Test tube 2 hopefully will uptake the DNA and get the kanamycin resistant gene and become transformed and be kanamycin resistant. Let's talk a little bit about kanamycin. There's a structure of kanamycin. It's a very beautiful chemical structure. And how does kanamycin work? So here is an E. coli cell, and you probably know that in an E. coli cell, the DNA gets transcribed to RNA. The RNA finds a ribosome, and that ribosome is going to make proteins which allow the cell to live. When kanamycin is present, it's going to enter the cell, and it binds with a small 30S subunit on the ribosome and basically blocks translation right at that very step. So no new proteins are synthesized and the cell dies. That's normally how kanamycin works. So how does kanamycin resistance work? There's our little cell, and we have a gene I mentioned on the plasmid. There's our plasmid in the E. coli cell. So the gene, of course, starts off as DNA, and DNA, of course, is transcribed to RNA, and that RNA finds a ribosome and makes proteins. So the kanamycin resistance gene is something called a phosphotransferase. And so it's a little enzyme, and that little enzyme is going to interact with kanamycin. So in this cell, the kanamycin enters the cell, and the phosphotransferase will actually phosphorylate the kanamycin and make it inactive. And so now we have an inactive drug, and the drug has no effect, and the cell continues to live. Now let's talk about the transformation procedure step by step and how it works. The very first step is we're going to add our plasmid DNA or the tris buffer to a couple of microfuge tubes. You can see 5 microliters is right there. It's a very small volume. Next, we're going to add the cells. These E. coli cells are called competent cells. And so what that means is they've been treated in a certain way that they will uptake DNA. So our cells have been treated with calcium chloride, and it's not entirely sure how it works, but what we think happens is that the calcium forms little holes in the outer membrane of E. coli, and that allows the DNA to bind and get taken up by the cell a lot easier. Like I said, the mechanism isn't entirely understood, but that's how we think it happens. So basically you've got leaky E. coli cells that are willing to take up DNA. Next, they're put on ice for about 15 minutes. This allows the DNA to kind of diffuse and soak around the solution and the E. coli to basically bind the DNA. The next step is something called a heat shock, and the heat shock is basically 42 degrees at 90 seconds. So they're going from a cold temperature to quite a warm temperature, and that is the step that induces the uptake of the DNA. It's very short. If you leave them too long, they'll die at 42 degrees Celsius. After that, they go on ice for 1 more minute just to give them a chance to basically take up that DNA and bring it inside the cell. At this stage, we need the cells to have a chance to be able to take the DNA, transcribe it to RNA, and make protein, so that phosphotransferase protein. So what we do is give them a little bit of food. That food is called LB media, so 400 microliters, and then what I do is I put it in an incubator for about 30 minutes. Sometimes I'll leave it longer. An hour is fine. In this case, I left it for about 40 minutes and incubate to allow the transcription and translation processes to occur so that they can obtain their kanamycin resistance. After that is one of the last steps in the procedure. We are going to take the cells and put them on plates with kanamycin and 1 plate without kanamycin. So you can see in the end we're going to have 4 plates. So LBK1, so LB is Loria broth, and K stands for kanamycin. Tube 1 was our control. LBK2, that was our experimental tube. We also have one called 1 no can, so that's a plate without kanamycin. That's another control plate. And then lastly, control culture, which is basically the culture from lab 8, the one that is already kanamycin resistant. So if you take a look at these plates, we have basically 1 experimental plate and all the other 3 are controls. Hopefully you've paid attention because you'll have to explain that in your lab report. Here's a brief video of me adding the culture to the agar plates. So 100 microliters is pipetted to the surface of the plate. Everything here is done aseptically, of course. I'm going to spread the culture around the plate with that bent glass rod. I'm not sure if it has a name. I call it a hockey stick, and spread it around the entire surface area of the plate. In the last step, we're going to incubate our plates at 37 degrees for about a day or 2. I will check after a day. If the colonies are not growing enough, I'll leave it a little bit longer. Once the colonies have grown, we're going to take a look at the results and count the colonies and enter your data in table 9-1. Here are the results. So I'm not going to explain what's in each of these tubes anymore. I think I've done that at least a couple of times already, but there are the results. You can see in plate number 1, there are no colonies. Plate number 2, there's a bunch. Plate number 3, there's growth all over the plate. And plate number 4, there is growth on the plate. So take a look at table 9-1, fill that out, and you'll have to answer the questions for the lab report. The Lab 8-9 short report. So here it is, the Lab 8-9 short report, the last short report of the semester. Yay! So for the short report, questions 1, 2, and 3 pertain to Lab 8, and the last one, question 4, pertains to Lab 9. So for question 4, what you're going to have to do is basically describe today's experiment. So what was it we were trying to do? What question were we asking? What were the variables, controls, and of course discuss the results. You're going to hand that in and make sure you do check the lab schedule for the due date. And then you're going to have one more thing to do for me for the Biology 107 lab, and that is a lab exam. So I will be sending out details in the next week about what to expect for the lab exam. Please check the lab schedule for the date of the lab exam. And then, yeah, so I hope you enjoyed these virtual labs. It really has been unfortunate. I know that students usually enjoy the lab exercises for Biology 107. They're a lot of fun. I enjoy doing them with the students and interacting with you, so it really is a shame you weren't able to do that. So if you do see me around, either on campus or in the community in the future, please say hi and feel free to ask me any cell biology or microbiology questions. I wish you all the best of luck for your final exams and whatever your future endeavors are.